My name is Dr. Erica Suosaari, and I am a science fellow with Bush Heritage in Hamlin Pool, working on the stromatolites. This paper was published in February in Nature's open access journal Scientific Reports, called New Multiscale Perspectives on the Stromatolites of Shark Bay, Western Australia. This paper takes an entirely new approach to stromatolites of Hamlin Pool. The reason we were able to take this new approach on the stromatolites of Hamlin Pool is because previous research has always been concentrated to the southeast margin of Hamlin. If you look at this ground truth map of areas we've examined in Hamlin, you can see that suddenly we're not restricting ourselves to the southeast corner of the pool, but instead we search the entire margin. As part of the investigation that we used for the Scientific Reports paper, we took 45 stromatolite heads from Hamlin, a number of pavement samples, over 100 sediment samples. We took single beam sonar from across the pool to build a bathymetry map, and basically spent months of just towing behind the back of the boat to examine the stromatolite and sediment types. This is a brand new model for stromatolite growth based upon the Playford model. We've identified that there's a difference in microbial structures in Hamlin Pool. Some of the microbial mats are just forming sheets. They can be laminated or unlaminated, but they're unlithified to weakly lithified, and they're not accreting upwards. These sheets are not building structures. Then, as you move into the intertidal and down into the subtidal, the microbial communities are actually building discrete buildups. They're accreting and building up and making the shapes that we know as stromatolites. In the upper intertidal, we have pustular mats that the dominant microbe is Entephilus. And Entephilus makes an unlaminated internal fabric. In the lower intertidal, we have smooth mats dominated by coccoid cyanobacteria. They make laminated structures. And in the subtidal, we also have coccoid-dominated cyanobacterial mats, but they're making kind of a jelly type of mat. This type of coliform mat makes a moderately laminated internal fabric. Another difference is that we find filamentous cyanobacterial mats are found dominantly in the upper intertidal, whereas coccoid cyanobacterial mats are found more towards the subtidal. Filamentous means they're filaments, they're like fingery, and coccoid are like they're little balls of microbes. This figure is about Entephilus. Entephilus is a coccoid cyanobacteria, and if you're American, sometimes you say that Entephilus. A comes from an old Aramic diagram, and it shows a living Entephilus top, so this is a living coccoid cyanobacterium, and as it degrades, how that shrinks down, and that's what the cement forms around.
What's happening in B?
Oh, it's just really showing you the coccoids and the dead coccoids, and you can see the dark dead coccoids.
Yeah.
Those are what the calcium carbonate is going to begin to precipitate on.
And what's C?
See, you can see the cement, the little white blobs of sediments that are being formed within this gel matrix.
And what's D?
D is this micrite, this calcium carbonate that's being precipitated around the dead coccoid cells within the organic matrix. This diagram beautifully shows the differences between the poorly lithified sheet mats and the microbial communities that are building stromatolites, so these lithified structures. So on the left-hand side, you can see smooth and pustular mats that are forming in the intertidal zone, upper intertidal. You've got just sheets of pustular mat and sheets of smooth mat, and that smooth mat is formed by a filamentous cyanobacterium called Microcoleus. This mat also has some schizothrix within it. Both are filamentous and they're not forming lithified structures. Then the pustular mat is also making these sheets in the intertidal, and it's dominated by Antiphysalis major and Antiphysalis granulosa, and they're both little coccoid cyanobacteria, and they make this kind of squishy bulbous mammillate mat. But then as you move deeper into the subtidal, you have another form of pustular mat, and this pustular mat, instead of just making sheet mats, suddenly the communities become more diverse, and that diversity is creating a lithified structure. I actually call this pustular mat crusty pustular, because if you feel a pustular sheet mat, it's that soft squishy material, but if you feel a pustular mat that's building a structure, it becomes a bit more jagged, a bit more rigid, crustier. And that has another cyanobacterium in its community that's not in the unlithifying sheet mat, and that guy is the filamentous cyanobacterium Dicothrix. Then as we move a bit deeper, we have another smooth mat, but instead of being dominated by filamentous cyanobacterium, this smooth mat is dominated by coccoid cyanobacterium, but it's still building this beautiful sheet mat. It often feels a bit crusty or a bit leathery. In the subtidal, the stromatolites are dominated by a coliform mat, which is also created by a coccoid cyanobacteria. This coliform mat often has a gelatinous feel to it. In our Hamlin pool study, we sampled 45 stromatolite heads. From the 45 stromatolite heads, we made 3 slabs. 2 we impregnated with epoxy and ground down and polished, so they're museum quality slides. 1 has stayed in Australia and 1 has stayed in the US. The center slab we cut into smaller pieces to make thin sections. So what this diagram is showing is that the top part of this stromatolite that looks green is just cement with not a lot of sediment incorporated. The center of the stromatolite, where the next thin section was taken from, has a lot of sediment in it. So you can see that that fabric is created by more of a trapped and bound mechanism. The next slide shows a different stromatolite taken from a different region of Hamlin Pool. The top image shows this clotted colloidal micrite and not a lot of sediment, but it's a totally different type of cement than the cement formed in the previous slide, which means that the mechanism of cement formation is different, which also means that the microbial community forming that type of cement is different. So within Hamlin Pool, we have a lot of different microbial communities, and we have a lot of different processes that's generating cement that's building stromatolites.